Final stretch of exam 2 material. So the last thing we're studying is the cerebellum. I just want to say real quick before we get started on it, a lot of people, and I think I've mentioned this before, call this exam the tract exam, and it does take up a good portion of the exam, but I feel like a lot of people neglect the cerebellum because it's the last thing in the exam material, and there is a decent amount normally of information on the cerebellum, and there's a lot of information that we cover about it. So don't put it off. Make sure you know all this stuff, and we'll go ahead and get started. So I have next to the rhombencephalon, I'm on page 91. I put up on my slide, and I also wrote it right under the big lettering on the page. Ron met Maya, if you can remember that from the first exam material. That was, so rhombencephalon is the primary brain vesicle, and it forms the metencephalon and myelencephalon. So just a reminder of that. Remember, mom met Maya, so you can remember which secondary vesicles come from that primary or rhombencephalon. And then it's listed as number 2 in your book. I also threw it up on my slide. It says, which are included in and then myelencephalon question mark, that becomes the medulla oblongata. And then the metencephalon becomes the pons and the cerebellum. So so what we're learning about in this section is these mature structures, medulla oblongata, pons, and cerebellum. So know which ones come from which secondary vesicles. I already kind of touched on it, but D is saying that this input is entirely subconscious. If you can remember with the spinocerebellar tracts that it's subconscious information, proprioception, and so forth. Hopefully that'll help you remember that in general, the cerebellum is all entirely subconscious. So make sure you know that. And then the end of the little paragraph under D says each cerebellar hemisphere influences the ipsilateral side of the body. So the diagram under that is just showing how the cerebellum on the left that's labeled, it might cross over to different sides of the CNS, but it is always going to end up influencing the same ipsilateral side. And then there's just a couple examples under on how that works, but I wouldn't worry too much about that. And now we're going to talk about the attachments. So I already talked about this a few times or a couple times, so hopefully you guys are all over this. The medulla oblongata is the most inferior part of the brainstem. Makes sense that it's going to be attached via the inferior cerebellar peduncle. The pons is the middle portion of the brainstem. It's attached to the cerebellum via the middle cerebellar peduncle. Midbrain is most superior, and it's attached via the superior cerebellar peduncle. So pretty simple concept there. Then we have the peduncle fiber distribution. So right off the bat, what I wrote in my book is I wrote pons over middle, and then you'll notice the pons, it says, or the middle says afferents only. So I just wrote in caps over that, like just over the whole little paragraph there, only. And that's how visually when I was studying, I just looked at that and I remembered, okay, pons, middle, only. So, and this is referring to the cerebellum. So it's afferents to the cerebellum only from the pons. You're not going to have any fibers that are going from the cerebellum to the pons. It's only from the pons to the cerebellum. And then the two on the other sides of that, so the inferior and superior, they're just like kind of flip-flopped. So the inferior is going to be afferents dominant, but there are some exceptions where efferents are present. And then the superior is the opposite of that. So efferents are dominant and afferents are still, there's a few exceptions where they're present. A way for me to remember which was which is I just drew arrows going up, I thought from going bottom up, and then that kind of helped me remember so the green one for the afferents and the inferior, if you look at my diagram, it's going in, or afferent, it's going up, and then the top one, if you just continue going straight up, it shows the superior part of the brainstem leaving. So that was just like a little trick that I used to remember that. Hopefully that makes sense for you guys. Didn't feel like typing out notes, so I just put a picture of my book up here. So for this section, we're talking about the vela. And the important thing to know, so we have a superior medullary velum and an inferior medullary velum. Big thing you want to know here is that childhood brain tumors most commonly happen in the superior medullary velum, and these are called medulloblastomas. Thing I want to point out is the name, medulloblastoma. So if someone reads this, they might think medulloblastoma, oh, it's a cancer or a tumor of the medulla oblongata because the word medullo is in there. But realize it's talking about medullary velum and not medulla. So don't get that confused. And just to point it out, I put a star in the bottom left. The obex, remember that is the kind of apex that opens into the central canal. You can see that right under the inferior medullary velum, and you see the arrow going down. So that's just another image to show where that's at. And it also labels the rhomboid fossa, so you can then again see how it lays on the backside of the pons and is the floor of the fourth ventricle. All right, this page has a lot of stuff going on, and as you can see on my chart, I wrote out memorize, and that's pretty much what you have to do, but we'll go through it too. I'm going to go through this diagram up here first. So this is going over the lobes of the cerebellum. The top one, it says it's the superior view, so looking down on it, you have our midline structure that's in kind of a pink, that's the vermis, and you can also see that from the inferior view. Then we have the anterior lobe, which is in orange. And then there's the posterior lobe, which you can see is the biggest lobe. So some students got tripped up when they got asked that on the exam previously, and my guess is it's because they didn't know this diagram, because it's not listed explicitly in the book, but just looking at this diagram, it's very obvious that the posterior lobe is the biggest there. And then we have a flocculonodular lobe. So the nodule is only on the inferior view. You can see it, it's in the middle. And then the two flocculus, or I don't know if they would be called flocculi for plural, they're going to be the two lateral structures. So together it's called the flocculonodular lobe. So we just talked about the lobes. This is where it gets a little confusing. So this is like physically parts of the cerebellum. So we have our lobes. We have, if you look at the chart, we have divisions, and then we have functions. So we have our lobes, but the cerebellum is also divided into its functional divisions, and that's what that first line is. So, for example, the first one, the vestibulocerebellum, is where you look at the function, where the information on posture, equilibrium, and balance goes. So, functional division is vestibulocerebellum. It deals with posture, equilibrium, and balance. And the lobe that it's in, or where it's located, this functional division, is the flocculonodular lobe. And then from there, you have to know the synonym. So vestibulocerebellum is also known as archicerebellum. It's going to get its input from the vestibular nucleus, which is kind of nice because it has the word in it. So vestibulocerebellum is getting information from the vestibular nucleus on posture, equilibrium, and balance. And if you're familiar with like the term vestibular apparatus, that's dealing with equilibrium. So hopefully that sounds familiar, and you can kind of connect that this equilibrium, balance, posture is all dealing with this vestibular stuff. This first one's kind of easy because the flocculonodular lobe is just one lobe and it's one functional division, but this next one, the functional division is located in multiple lobes. So that's, I think, where it gets kind of confusing. So we have our spinocerebellum, that's the functional division. I'm going to jump to the bottom. That is receiving input on muscle tone, trunk, and limb movements. Its inputs that are sending this type of information are going to be our spinocerebellar tract and cuniocerebellar tract. That's a new tract that we don't learn anything else about in this class. What's kind of nice is that our input, one of them, the spinocerebellar tract, is going to the spinocerebellum functional division. So that's kind of nice. So maybe the one that he'll ask you about is the cuniocerebellar because it doesn't kind of give it away. And then when you look at the lobes, it's located in the anterior lobe, the vermis, and the medial posterior lobe. So here's a way to remember that. If you look at the top diagram that I posted, you'll see I drew, it kind of looks like a T around the superior view, and I call it T-spine. So if you look at what it's covering, it's got the orange anterior lobe, it's got the medial vermis, and then it extends out a little bit just to include the medial posterior lobe. So if you remember to think of the T-spine around it, and you know what this diagram looks like, then that gives away the lobes that are a part of the spinocerebellum functional division, or where the spinocerebellum functional division is at. So that's a good way to remember that. And then I call it T-spine because of spinocerebellum, or it's coming from the spinocerebellar tract. And then from there, you just have to remember the cuniocerebellar tract is also an input for that. And then for me, when I was studying, I remembered vestibular, so I thought posture, equilibrium, balance. So then when I thought spine, I thought what's something that goes along with it, and it just kind of made sense to me that muscle tone, trunk, and limb movements came from that, because the trunk is kind of where your spine is at. I don't know. Hopefully that helps you guys. And the next functional division is our cerebrocerebellum, also known as neocerebellum. Don't forget, he can use either the synonym or the division to ask the question, so you need to know the synonyms. So this one is located in the lateral posterior lobe. That's the last of the lobes that we've covered. Inputs are from the corticopontocerebellar tract, and we don't talk about that elsewhere besides with the cerebellum, so you don't need to know like any details about that. Just know corticopontocerebellar goes to the cerebrocerebellum. And the function that we're dealing with here is planning and coordination of skilled movements. So how I remember this, the cerebrocerebellum, if you break that down, cerebro stands for cerebrum, and we haven't talked about the cerebrum yet, but we've talked about the corticospinal tracts, and those come from the cortex of the cerebrum, kind of like how if you look on the chart, it says corticopontocerebellar. And what does that deal with? Initiating fine skilled movements, right? So that's how I connected the cerebrocerebellum is dealing with the planning and coordination of skilled movements. So it might be kind of a stretch for you guys, but it helped me remember it. Just a couple other things on this chart I want to point out. So I labeled, and you might want to do this too, we haven't talked about Purkinje neurons yet, but next to, on the very right side of the divisions, I wrote, this is where Purkinje's leave. So I would write that as a reminder, so when you go back and study, it makes a little more sense when you see it again. And then at the bottom, and this is in your book, but I just wrote it because I wanted to have another place to see it and put all the information together. Purkinje axons are going from these functional divisions to what I have in red there. And I crossed out the vestibular nucleus in the medulla oblongata, but you can put that. That is true. I don't know why I crossed that out. So from the start, vestibulocerebellum. What's nice about this one is the input, it's receiving input from the vestibular nucleus, and then the output, where the Purkinje axons go from there, is right back to the vestibular nucleus. So that's pretty easy to remember. From the spinocerebellum, Purkinje axons are going to leave to the interposed and fastigial nucleus, and then from the cerebrocerebellum, they're going to go to the dentate nucleus. So a lot of new words. I have a pretty easy way to remember these, so don't freak out just yet. I'm going to skip page 108. That's just showing an unwrapping of the cerebellum. Internal cerebellum is next, so a couple things I want to point out on this page. White matter collectively is called corpus medullare. Know what that means. Just a reminder that there are 6 cerebellar peduncles, so we talk about the inferior, middle, and superior cerebellar peduncles, but they are bilateral, so there's 2 of each. And then extensions of white matter, looking at number 4 on the page, are called arbor vitae. If you flip back to page 107 and look on the right side where that diagram is, it shows a mid-sagittal cut with the arbor vitae, or the extensions of white matter. So just to point it out, don't get arbor vitae and corpus medullare mixed up, because they're both kind of like strange words. So they mean different things. Moving to gray matter, externally, don't forget fissures means deep and sulci means shallow. How I remembered that was SNS for sulci and shallow. And we're going to talk about the neuron cell types on the next page. So I'm going to go through this how I usually go through it in PASS, where I kind of just break down the diagram here. And if you understand this, then you'll understand everything in the next couple of pages. So this diagram is showing the cerebellar cortex. So the cortex is the most outer part of the cerebellum. This is the gray matter. Page 109 at the bottom said most common neuron cell types, and then it went into the top of page 110. And looking at this diagram, we're going to cover all of that. So just looking at the diagram in the cortex, there's 3 layers. We have starting from the bottom, the granular layer, then the Purkinje layer, and then the molecular layer. The molecular layer is the most superficial layer, and I wrote up at the top superficial pia. So the pia, or if you remember, that's the most internal layer of the dura, is going to be right next to that. So then the granular layer is going to be the most internal layer. Think of these layers, at least the bottom 2, as being named for the type of cells that you can find there. So the granular layer has the granule cell, and I highlighted it to kind of connect it. The Purkinje layer has the Purkinje cell. The molecular layer has the Purkinje dendrites, and we'll go into that in a second. So what we're describing here is we're going to go through the axons that bring information to the cerebellar cortex, which is what we're looking at on this diagram, and which layers, and then from there, what happens. So I'm going to start with, and I have it at the top highlighted in like a darker pink, mossy fibers. So look, if you look at the mossy fiber I've labeled, excuse me, the mossy fiber goes to the granular layer. It's synapsing on the granule cell, and you need to know this. And I have the granule cell in another type of pink. The granule cell is then going to go up to the molecular layer, and it's going to synapse on spined branches of Purkinje cells. So that's one way we get input to the cerebellar cortex. The other way is a little bit simpler. We just have our climbing fibers that I highlighted in green, and they just go straight up to the molecular layer. So the climbing fibers don't stop, whereas the mossy fibers don't make it all the way up to the molecular layer. They synapse on the granule cells, which then go to the molecular layer. The climbing fibers synapse in the molecular layer on smooth branches of Purkinje cells, and they're going to release aspartate. I have the path of both of those written out at the top of my page, which is why I just took a picture of it. So I recommend you guys doing that as well. It takes everything we're looking at on the diagram and puts it into writing. If you look at the mossy fibers that go to the granule cells and then spine branches, those are going to release glutamate on the Purkinje cells. I know we talked about glutamate for sure, and I think we talked about aspartate too in exam 1 material. Remember we said glutamate was excitatory because a mate's glutes are excitatory, and then aspartate was extremely excitatory because it's got a more, I guess, aggressive term for glute there, aspartate. So remember, glutamate is excitatory, aspartate is extremely excitatory. And I kind of follow, I'm going to come back to that, but I kind of follow a story with remembering whether our cells are synapsing on spined or smooth branches. So I'm going to start with the climbing fibers. It's our green one on the page. Climbing fibers, this is how I tell this story to myself when I'm studying. Climbing fibers have a smooth ride up to the molecular layer because they don't synapse on anything else. They go right up. So I consider that a smooth ride, which then tells me they're going to synapse on smooth branches of the Purkinje cells. And because they have a smooth ride and they don't have to stop, they're going to release something really excitatory, aspartate. So that's how I remember that one. And then going from that helps me figure this story with the mossy fibers. So mossy fibers don't have a smooth ride because they don't make it to the molecular layer, they have to synapse on the granule cells in the granular layer, which then go up to the molecular layer. So it's not a smooth ride, which is what tells me they're going to synapse on spined branches. And I kind of tried to draw some little spines in the pink to help you visualize that. Since the mossy fibers to the granule cells don't have a smooth ride, they're still excitatory, but they're not going to be super excitatory. So they're going to release glutamate rather than aspartate. So if you can remember that, that was super easy way for me to remember the pathway of both of these and what they're releasing. One thing I didn't write, granule cells or mossy fibers are also going to release glutamate on the granule cells. So with the pathway that I have in the pinks on the right, it's glutamate being released at both dendrites. From there, so we synapse on the dendrites in the molecular layer, and then the cell bodies of the Purkinje neurons are going to be in the Purkinje layer. And then they're going to leave, and if you look at the bottom right of this diagram, it says to deep cerebellar nuclei and lateral vestibular nucleus. So that's where the Purkinje neurons go. And those are going to release GABA, which I have written under number 6, and I would write that as well if I were you. So Purkinje fibers are releasing GABA, and that is an inhibitory chemical. So number 5, these pathways we're talking about to the Purkinje layers are releasing excitatory stuff, because otherwise the Purkinje neurons wouldn't fire, right? And then when the Purkinje cells leave, those are going to be inhibitory, always. So Purkinje fibers are always releasing GABA, always inhibitory. And then we'll talk about the deep nuclei that they go to in a couple pages here. And I just want to point out, I wrote it on the right side at the end of number 6 there. They're taking messages away from the cerebellar cortex, not the cerebral cortex. So just look out for the wording. It's cerebellar, not cerebral. One more thing with the climbing fibers, if you can remember when I said we'd get back to it, when we talked about the medulla oblongata, we said the inferior olivary nucleus sent climbing fibers to the cerebellum. So that's where we're seeing this now. And I wrote it next to the climbing fibers up on my slide or my page, so you can kind of add that to the pathway. So the climbing fibers that are going to the molecular layer are coming from the inferior olivary nucleus. And then further down on the left, I have written in red. This can trick you if you're not reading carefully. So let's talk about the differences in these two bullets. So Purkinje cells are the efferent from the cerebellar cortex. They are the ones leaving the molecular layer, and if this diagram is the cortex showing you the layers in the cortex, we have arrows showing that it's leaving that. Under that, it says cerebellar outflow ? So the difference here is not leaving the cortex, but leaving the cerebellum totally. So the Purkinjes aren't leaving the cerebellum, they're just leaving the gray matter of the cerebellum to get to the deep nuclei. And then the deep nuclei are what is the efferents from the cerebellum. I hope that makes sense. I'll try to remember to bring it up again when we look at the other diagram on this stuff. I'm not going to go through page 111. It pretty much puts in words everything I talked about on this page and wrote out in red. So read through that, and I would highlight any of the things that we talked about. It puts in words whether it's on smooth or spined dendrites and inhibitory versus excitatory chemicals and all that. So I'm not going to read through that again. I do want to talk about the very bottom of page 111. It asks, what were the 2 fiber types that reach the cerebellar cortex? So here we're seeing again the term cerebellar cortex. So looking at this diagram, the 2 fibers that reach the cortex, which is the diagram, right? The granular layer is the most internal part of the cerebellar cortex. It's going to be our climbing fibers and mossy fibers. This is important because that question was asking what reaches the cerebellar cortex. There's another way that this question can be asked that will change the answer, and that's which two fiber types reach the molecular layer. That's going to change it because if you look at the diagram, the mossy fiber doesn't reach the molecular layer. It reaches the granular layer only. It's only reaching the cortex on the internal layer, and then the granule cells go up to the molecular layer. So just going to repeat it one more time. Which fibers reach the cerebellar cortex? That's going to be climbing and mossy fibers at the bottom. Which two fibers reach the molecular layer of the cortex? That's going to be looking at the top. So climbing fiber still and granule cell. So I ended up typing out what happens from start to finish with the mossy and climbing fibers. So I color coordinated it to the previous slide with the diagram. So go through that. One thing I didn't mention, we talked about how the climbing fibers are coming from the inferior olivary nucleus. The mossy fibers are going to be pretty much everything else we talked about on the chart with the functional divisions. So in your book on page 112 at the top, it covers saying that mostly these mossy fibers originate from spinocerebellar and corticopontocerebellar sources. So pretty much covered all this, but I just wanted to have it all on one page in writing so you could take a screenshot of that. If you know the order and you study that, then questions like where do Purkinje cells go will be super easy. Purkinje always goes to the deep cerebellar nuclei, and that's listed through page 112. I'm not going to read through that, but you guys should do it yourselves. I put pretty much everything on this page that you need to know. Don't forget, though, we talked about it before, some Purkinje fibers go to the vestibular nucleus. So don't forget that. It's still going to release GABA. They're always releasing GABA, which is inhibitory. You've probably noticed there's a ton of stuff going on in here. It's showing the brainstem on the left, and then we have the peduncles attaching the brainstem to the cerebellum. So what I have highlighted are the deep nuclei, which we have at the bottom of page 112. I think I'm just gonna go through and add on to this for each slide. I think that's the easiest way. If you know this diagram, it makes this information a whole lot easier rather than just memorizing all these words and where everything is going and all that. So just as a reminder, the deep nuclei are in the middle, or the central nuclei. That's where the Purkinje neurons go. So you look and you see from the outside of the cerebellum, or the cortex, I have it highlighted in yellow, that's where the Purkinje neurons are leaving, and they're going to the deep nuclei from there. They're releasing GABA, which is inhibitory. I know I've said it like a million times by now, but it's super important that you know it's releasing GABA, and that's inhibitory on the deep nuclei. So I'm going to start with dentate, and if you think of it this way, it's super easy to remember this. Dentate is the largest and most lateral, so I think of that outside portion that you're looking at as the most lateral. Next we have emboliform, and then globose, and I put those the two similar colors because collectively they're referred to as the interposed nuclei. So make sure that you know interposed is emboliform and globose. And then last, the most medial, and it says that in your book as well, is fastigial. It's the most primitive and most medial. So make sure you know that, but it's easy just by knowing the diagram to realize that it's most medial. So a way to remember the order of these, I learned from my board's review, is flowers grow every day. So that's going from medial to lateral. So F stands for fastigial, G globose, E emboliform, D dentate. So quick, easy way to get the order down. It is the closest to the vermix because the vermix, remember, was that midline structure in the cerebellum. Okay, so I realized the image that I originally had up had that some of the arrows were weird and didn't make sense, so I ended up just putting mine up. So, like I said, there's a lot of stuff going on here. We're just gonna go through at the top of page 113, the little chart there, the little flow chart, one by one, and hopefully it'll make sense by the end. So try to just focus on what I'm talking about, and I'll point you to where I'm at, and then at the end you can kind of go through and read through this diagram all together. So we have our fastigial globose emboliform and dentate labeled. Those are the deep nuclei. And then on the outside of the cortex, that's where our functional lobes are. So the first one I have labeled is vestibulocerebellum. It's the green one that I highlighted. So if you go around, so I'm kind of going clockwise, we have the vestibulocerebellum, and then we have the spinocerebellum to the right. I have it in pink. And then below that is cerebrocerebellum. So a way for me to remember these is I just said they were backwards alphabetical in clockwise fashion. So it just goes V, S, C, and it goes around. So that's how I remember that. Do you guys really need to know that? Probably not, because you won't be labeling this on an exam. But knowing it is going to help you kind of keep everything straight when it comes to where all of these neurons are going. So starting with vestibulocerebellum, if you remember when we had that, you might want to turn to the chart on page 107. So this is the functional lobe that's in the flocculonodular lobe, dealing with posture, equilibrium, and balance. We said the input was from the vestibular nucleus, or Dieter's nucleus. And if you look, it's labeled there at the kind of bottom left of the diagram, right in the medulla oblongata. So we're going to have our axons coming from that, going up, synapsing in the vestibulocerebellum. And then from there, if you can remember when we talked about the chart, we said this one's easy because our Purkinje axons are leaving and going right back to where they came from. So we're going from vestibular nucleus to the vestibulocerebellum, and then we're just going right back to the vestibular nucleus. So that one's pretty easy. Then next we have the spinocerebellum. So this, if you can remember from the chart, we had our inputs coming to the cortex from the spinocerebellar and cuniocerebellar tracts. So that comes in, and those would be the examples of our mossy fibers that are going to the granule cells and then would make it to the molecular layer. And then what you see highlighted on the image is the Purkinje cells leaving. And those are going to go to interposed and fastigial nucleus. So the three most internal dentate nuclei. I know my highlighting just kind of looks like a blob there, but it's the three most internal ones and not the dentate on the outside there. So, and that's written in your book too. Here from that chart on page 111 or whatever it was, corticopontocerebellar are going to the cerebrocerebellum. Those are an example of the mossy fibers again. And then from there, they're going to go to the dentate nucleus. So that's what I have highlighted, the Purkinje neuron going to the dentate nucleus, releasing GABA, which is inhibitory on the deep nuclei. So deep nuclei, once again, are receiving inhibitory GABA. So everything we've talked about has been mossy fibers coming into these functional divisions and releasing glutamate on the Purkinje fibers, and those going to the deep nuclei or the vestibular nucleus. What we haven't talked about are climbing fibers yet. So I have it labeled in orange down there, the inferior olivary nucleus, and it's written under this little flow chart on page 113. These are non-cerebellar influences on the deep central nuclei. So our climbing fibers that are coming from the inferior olivary nucleus go to all four nuclei, and then if you can remember, they're going to release aspartate, because we said climbing fibers have a smooth ride. They're going to synapse on smooth dendrites of the Purkinjes and release aspartate, which is extremely excitatory. So that's just one of the non-cerebellar influences on the deep central nuclei you need to know about. Before you start reading this paragraph that I put up, I just want to say a couple things on remembering. If you know this chart or this diagram, the vestibulocerebellum, I feel like we hit pretty good, and that's one of the easier ones. The input and output is the same to and from the vestibular nucleus. The cerebrocerebellum, if you look at the diagram, you'll notice it goes to what's closest to it. So if you know the dentate is the largest and most lateral, the cerebrocerebellum is right there and goes right to it. So that helped me remember that. And then from there, the spinocerebellum goes to what's left. So the three that are internal to the dentate is what spinocerebellum goes to. So I hope that helps and I didn't just confuse you a little more, but I wrote out this paragraph a few trimesters ago and put it on the files page that goes over the outputs from the deep nuclei, which you definitely need to know. And that's at the bottom of page 113. So I noticed a pattern in where they go, and I figured rather than me talking and you trying to follow along to my voice, I just paste this on there for you guys to look at. So pause and look through that, and then we'll move on to, I think, the last page of this exam information. One thing I do want to mention, we already talked about it, but just want to say it again since we're here. It says under the deep nuclei, 4A says, in essence, this is the efferent cerebellar outflow. So don't forget, this is where we have efferents leaving the cerebellum. That's different than leaving the cerebellar cortex, which would be the Purkinje neurons. Another thing, if you didn't notice, this middle section where you can see the pontocerebellars, this is the cerebellar peduncles. So the brainstem is obviously on the left, the cerebellum is on the right, and then those are the peduncles that are attaching the brainstem to the cerebellum. So if you can remember when we talked about that chart on the afferents versus efferents, you only see neurons going to the cerebellum from the middle peduncle or the pons. We said the medulla oblongata on the bottom, or the inferior peduncle, was afferents dominant, some efferents. So you can see it's mostly going into the cerebellum from the medulla oblongata. But the exception is going to be where you see that, and we haven't talked about it, I have it typed out, but the fastigial nucleus is going out to the vestibular nucleus through the inferior peduncle. So that is an example or an exception of where the efferents are present, but afferents dominant there. And then the superior peduncle is going to be efferents dominant, and you can see the dotted lines are the efferents leaving the cerebellum. So you can see there's a ton of dotted lines, but there are some exceptions where we do have afferents, and a good example of that would be our anterior spinocerebellar tract. If you remember when we talked about ass backwards tract, it was AS stood for anterior spinocerebellar, superior cerebellar peduncle is what it goes through. So that is an example of where we have the afferent, and you do need to know whether these are traveling through inferior versus superior versus middle. And it's nice just to know the rule that the pons or the middle cerebellar peduncle, we are only going to have afferents going to the cerebrum. Sorry, cerebellum. I don't know if I was calling it the cerebrum this whole time, but we're talking about the cerebellum here. See, even I get a little mixed up and don't think straight when we're talking about the two. The words are so similar. All right, next we have cerebellar disorders. So I'm just going to highlight a few. First is we've got B is equilibrium disturbances. So hopefully you can connect when we talk about equilibrium, what functional division are we talking about? Probably the vestibulocerebellum. Remember we said vestibulo, think vestibular apparatus, think equilibrium, posture and balance. So this is also termed flocculonodular syndrome, and that's in your book as well. And then from there, hopefully, again, you're connecting that the vestibulocerebellum was the functional division that is in the flocculonodular lobe. So that kind of adds up. Next is ataxia. If you don't know what this means yet, it's a lack of order or coordination. So our coordination was coming from the neocerebellum. If you remember, the neocerebellum is also the cerebrocerebellum. So be aware of where that is coming from. So if you have a lesion of the neocerebellum, this is what your results are. And it says in the book, here one might stagger to the side versus falling backward in flocculonodular syndrome. So with flocculonodular syndrome, your equilibrium is off, your balance is off, so you're going to be falling. Whereas with ataxia, you don't have the coordination, so you're kind of just like staggering. You're not necessarily falling. Hope that makes sense. And then we have intention tremor. We've talked about this, I think we've talked about this before. So it's when you're intending to make a movement rather than, it says it's unlike Parkinson's, where with Parkinson's we learned that that's a resting tremor. So intention tremors happen more with MS, and this is influenced again out of the neocerebellar lobe. Hopefully that makes sense because the neocerebellar lobe is in control of planning and coordination of movement. Then we have nystagmus, which is a repetitive jerking movement of the eyeballs. I'm not going to read off this paragraph here, but I will mention, since I didn't type it in that description that I posted on the last slide, the fastigial nucleus has fibers that go to cranial nerves 6, 4, and 3, so that LR6, SO4, all over 3, those are the voluntary eye muscles. So then putting that together, you could see how having something wrong or a lesion can affect the eyeballs, and then know the term that it's referred to as nystagmus. So know what these words mean, know what's being affected, and that is all I have for you for the rest of this exam material. So good luck studying.